Warm good afternoon to all. Henry Ford says, coming together is a beginning. Keeping together is progress. Working together is success. With the blessings of Almighty God, I am happy to join with you on the fourth day of International Faculty Development Program organized by PG and Research Department of Microbiology, Idhaya College for Women, Kumbakonam. Now, I invite our HOD, Dr. R. Krishnaveni, convener and organizing chair of the FDP, to deliver the inaugural address.
Good evening to all. I invite you all today to attend the session by Dr. J. Prakash Sagaya Leon, Assistant Professor, Government Arts College for Men, Krishnagiri, debuted from Annamalai University, Chidambaram. He delivered a topic in the title of Toxicological Impact on Aquatic Biology. Now I invite Mrs. T. Megla ma'am to deliver welcome address and introduce the guest.
Thank you, ma'am. Welcome to today. Another day, another chance. Feel free to change. The presence of God with us here is today. We gathered to get the chance to change. I am privileged to deliver the welcome address of today's session of International Faculty Development Program on the topic Toxicological Impacts on Aquatic Biology. A successful woman is one who can build a firm foundation with the bricks others have thrown at her. I am glad to welcome our principal, Reverend Sister Dr. Eugene Amala, who is a successful woman to guide us in the right path. One of the greatest gifts a person gives another is support. It is my pleasure to welcome our Secretary, Reverend Sister Claudine Oroke Mary, who supports us in all our efforts. Courage, encouragement, and hope are the two most powerful qualities. I am happy to welcome our Administrator, Reverend Sister Andoni Amal, and Vice Principal, Reverend Sister Dr. Vimali Mary, who encourages and gives hope to us in every step we take. Power is gained by sharing knowledge, not by holding it. It is my great pleasure to welcome and introduce our today's presenter, Dr. Prakash Sahaya Leon, who is going to share his knowledge on toxicological impacts on aquatic biology. He is working as assistant professor in Government Arts College for Men, Krishnagiri, who is working in deputation for Zoology Department of Annamalai University, Chidambaram. He has completed MSc, B.Ed, MPhil and PhD and specialized in the field of toxicology. He has guided 4 PhD scholars and 7 MPhil students. He has published over 25 research papers at over 35 workshops and conferences. He has also organized 8 conferences and completed a research project funded by UGC. I extend my warm welcome to our resource person. Welcome you, sir. Every day may not be a good day, but there is good in every day. With this quote, I welcome all the participants, who includes professors, associate professors, assistant professors, research scholars, and students of various disciplines to make this day as a good day. Once again, I welcome you all. Now, I hand over the session to our resource person. Dear participants, kindly mute your audio for the seamless conduct of the session, and you can post your doubts in the chat box at the end of the session.
Okay, I start the program.
Sir?
Okay, I'll start.
Okay, sir. Thank you, sir.
Good afternoon to all of you. I'm Dr. Leon. First of all, I would like to thank my heartfelt thanks to convener, principal, Reverend Sister Dr. Eugene Amala, Idhaya College for Women, Kumbakonam, and also my sincere thanks to the organizing secretary, Dr. or Krishnaveni, Assistant Professor and Head, Department of Microbiology, Idhaya College for Women, Kumbakonam. Also, I thanks to all the organizing committee members and the professors and the assistant professor, associate professors, and scholars, students, those who are on the online program. Thank you, all of you. Today, uh, the topic which is known for me is actually toxicological impact on aquatic biology. So, aquatic resources are valuable natural assets for a human. They provide generous long-term benefits. These benefits can be direct financial ones. They provide employment, profit, and money savings. Example, the seafood industry provides jobs for commercial fisheries. Overuse of insecticide, pesticides, metals, and fertilizers are become very toxic to aquatic organisms. Pesticides are big business. Over £1 billion of pesticides are used in various countries per year. Reason why we back up these chemicals. It is relatively useful, easy to apply, generally cost effective, only practical method of control in some situations. However, the benefits of pesticide are not derived without consequences. Pesticide must be used with great care so that the health of humans, animals, and the environment will be protected. The production of pesticide started in our country is 1952. India is now the second largest manufacturer of pesticides in Asia after China and ranks 12th globally. The main use of pesticides in India is for cotton crops. It is near about 45%, followed by paddy and wheat. How pesticide works? First, toxicity. It means how poisonous the pesticides are. Some pesticides are extremely toxic, whereas others are relatively non-toxic. Exposure time. When you see the exposure time, it is calculated how long the animal is in contact with the pesticide. A brief exposure to some chemicals may have little effort on field, whereas longer exposure may cause harm. When you compare the dose rate, when you discuss about the dose rate, the quantity of pesticides to which an animal is subjected orally, dermally, or through inhalation. A small dose of more toxic chemicals may be more damaging than a large dose of a less toxic chemicals. How doses can be measured? The doses can be measured as the weight of toxicant per unit kilogram of the body weight. It is expressed as mg pesticide per kg of body weight. If the concentration of toxicant in the water or food supply, it is expressed as parts per million or ppm. When some types of when you discuss about some type of toxicants, first we have to say about the carcinogens. These are cancer-causing chemicals. Usually, cigarette smoking one of the cause for this. Then we come for the mutagens. When our organisms are exposed to a mutagen, it mutates their DNA, leading to cancer and other disorders. Also, even X-rays. Teratogens. These chemicals cause birth defects during development in the womb, mainly cause harm to unborn babies. Even a single dose is powerful enough to cause severe birth defects. And allergens. Chemicals that stimulate overactivity in the immune system, for example, pollen and dust. And neurotoxin, chemicals that attack the nervous system. This includes heavy metals like lead, mercury, and pesticides. Neurotoxins can lead to symptoms like slurred speech, loss of muscle control, and even death. Some common pollutants, for example, organic pollutants, inorganic pollutants, pesticides, biological agents. These are the common sources for toxic. The common toxic metals, if you see, they are the heavy metals like mercury, zinc, arsenic, copper, lead, nickel, chromium, cadmium, arsenic. These are the common toxic metals in this world. So, how these toxic metals are influencing the human activities or in the human being? Mainly, it is influenced the age, influence of age. It is influencing the age. For example, see, compared to adults, very young children are often more susceptible to chemical toxins. This is because of greater inhalation, volume, and gastrointestinal absorption rate. Also, it is due to greater permeability of the intestinal epithelium and immature detoxification into systems. And influencing of sex. See that males are generally more susceptible to renal toxin and the females to liver toxin. The causes of the difference response between males and females have been related to differences in a variety of physiological processes. Example, females are capable of additional excretion of some toxic chemicals through menstrual blood loss, breast milk, but they experience additional stress during pregnancy, delivery, and lactation. We should know something about the aquatic biology. What is this aquatic biology? Aquatic biology deals with the biology and the ecology of um, environment. Now see that importance of aquatic ecosystems. What is the main importance of this aquatic ecosystem means? It is supporting for recycling the nutrients, and it is supporting for purifying the water and recharge the groundwater, provide habitats for wildlife, used for human recreation, tourism industry. It provides a food for people, and more than 50 millions of people find a source of income with the help of this aquatic ecosystem. And health benefits of the eating fishes. For example, the fish is the main source of rich amount of nutrients. Most of the people they are using the fishes. They are having the fishes as their food. For example, high in important nutrients, the fish contain high nutrients value, nutritive value. It may lower the risk of heart attacks and strokes. The omega-3 fat is especially important for brain and eye development. It is boosting the brain health, prevent and treat depression, a good dietary source of vitamin D, and it is reducing your risk of autoimmune diseases like diabetes 1, and help to prevent asthma insurance, protect vision in old age. Fish may improve sleep quality, delicious and easy to prepare. So these are the advantages we are having while consuming the fishes. Then just we are entering into the stress and aquatic ecosystem. The human population is giving more stress on this aquatic environment. How these aquatic organisms are exposed in this aquatic toxic environment. For example, direct absorption through the skin by swimming in pesticide contaminated waters, and direct uptake of pesticides through the gills during respiration, drinking pesticide contaminated water, feeding on pesticide contaminated prey. So these are the main cause for the aquatic organisms are exposing under this toxic chemicals. And the repeated exposure of certain pesticides may cause reduce the egg production in fishes. The fish population and the fish population is totally reduced because of the toxic chemicals. And hormonal changes is occur in the fish and decrease the body weight of the organism. And it is also lower the resistance to the diseases. A lot of problems fishes are getting, and not only fishes, even aquatic organisms which is living in aquatic media are getting affected. See the pictures which is given on the screen. It is stressed on aquatic ecosystem. The first picture you see, that is because of the toxic substances which is mixed with aquatic media. The fishes are, when the fishes are exposed to the toxic, they are dying. And the second picture you can see, that is there is an oxygen demand in the water. Because of that, the demand of oxygen, the fishes they are unable to breathe inside the water and they try to come out. So these are the problems. Not only fish, even the living organism which is living in the aquatic media, everyone is getting day by day, and daily if you see, lot of number of thousands of living organisms are dying because of the aquatic toxin. And some research work conducted in various places by us. by me and our team. This is on the screen. It is in anomaly university. When I was working in anomaly university, we have done a lot of work. And now presently I am working in the government arts college. There also we are doing a lot of research. The research mainly focused on the aquatic toxicology. And these are the setups I have done, I have made. And next one, some of the work what we have done that I am going to share with you. First, see the effect of perimeter copper on freshwater fish, Oriochromis. The test, the experimental metal actually we have selected is a copper. The test animal we have selected that is a freshwater fish, Oriochromis mosambica, it's a tilapia. What are the tests we have conducted on this copper? Medium lethal concentration of the study conducted for 96 hours, means that is LC50, and the sublethal study conducted for 30 days at various sublethal concentration of 1 by 10, 1 by 20, and the biochemical analysis of protein and amino acid, gill and liver of the fish, Oriochromis mosambicis, are conducted, and histopathological alteration in gill of fish, Oriochromis mosambicis, exposed to various sublethal concentration of copper for 30 days observed. So like that, in the result of this study, here you can see when you do the biochemical work, the result of protein level in the gill and the liver, you can see on the screen. When the gill is exposed in the toxic chemical, the protein level in the gill is comparing the control group, it is reduced. See, this is the control group, this is the treated group. Okay, group 2 and the group 3 are treated one. While comparing the control group, the protein level is decreasing in the gill. Likewise, the liver also, the level of the protein is decreased while comparing with the control. So, when you go for the amino acid, we have conducted the test in amino acid also. When you discuss about the amino acid, the gill, the level of amino acid is increased while comparing with the control group. Okay, and the liver also, the level of the amino acid is increased while comparing the control group. Okay, control and the next one. See here, when you discuss about that, what outcome from this? In this investigation, the level of protein decreased in the gill and the liver, whereas the level of amino acid increased at all the groups of exposure when Oriochromis was exposed to sublethal concentration of copper. The depletion of protein under the stress of copper toxicity observed in different tissues of Oriochromis indicates the proteolysis, suggesting that protein were utilized to meet the excess energy demands imposed by the toxic stress. The reduction in protein content in the present study indicates that the tissue protein undergoes proteolysis, resulting in the production of free amino acids. So these are the problems. These are the things we analyzed. Next, from this we are coming to the conclusion. The present investigation concluded that the heavy metals copper induced biochemical and histological alteration and caused a significant metabolic effect on the physiological consequences. These biochemical parameters offer a rapid and sensitive means of monitoring towards the impact of copper on aquatic.
No, ma'am.
Biochemical analysis of proteins, lipid in gill liver of the organisms we have conducted this, and hematological observation also we made it. Means RBC, WBC, hemoglobin. So like that, different hematological parameter study also we made it for 21 days. Here, the result of this study, you see the graph. Here also the total protein, level of the total protein, while comparing the control group, it is decreased in different various concentrations. Maybe it's a low concentration or high concentration. Okay, whatever it may be, that while comparing the control, the level of the protein is decreased. This is not only gill, even liver also the same. While comparing control, what you call the level of the protein is decreased. Next, we go for the lipid. The lipid level also, you see that lipid level also, while comparing the control, it is decreased. Okay, gill now even liver also. Gill and the liver, both the organs while comparing that control the lipid level is decreased in the fetal level treated. And hematological parameters, while discussing about the hematological parameters, they see the RBC level. The first graph is giving the RBC level. The RBC level is decreased. While comparing with the control, the treated group is getting less RBC. And when you come for the WBC, but the WBC level is increased. While comparing the control group, the WBC level is increased in the treated group. Then we come for the hemoglobin percentage. When you come for the hemoglobin percentage, while comparing with the control, the treated group are reduced. So while discussing this, what the reason for this? The elevation of the protein level in the treated group. That's the reason. The protein in gill and liver decreased. Okay, the protein level is decreased in the gill for 21 days. The decreased level may be due to blocking of the metabolism of amino acid, thereby preventing cells from synthesizing protein. A reduction in the protein content may occur. Protein undergoes proteolysis, which results in an increase in the production of free amino acid. Then you go for the lipid level. The lipid content is decreased in the gill and the liver of Oreochromis niloticus exposed to sublethal concentration of sodium chloride. The decreased percentage of lipid due to inhibition of lipid synthesis by sodium chloride. Decreased level of lipid in tissue indicates that lipid hydrolysis might be accelerated to deliver energy to overcome toxic stress. When you discuss about the hematological study, the total RBC and hemoglobin content, RBC and hemoglobin content were found to be significantly decreased, and WBC counting is significantly increased after exposing to sodium chloride. The decreased erythrocyte count observed in the study this way may be due to the disruptive action on erythropoietic tissue, which in turn affected cell viability. The increase in WBC count can be correlated with an increase in antibody production, which helps in survival and recovery of the fish exposed to sublethal concentration of chemicals. Decrease in hemoglobin values may be due to the lysis of erythrocytes. Now, conclusion. From this study, we concluded that the present investigation concluded that the sodium chloride induced biochemical and hematological alteration and caused a significant metabolic effect on the physiological consequences. These biological parameters offer a rapid sensitive means of monitoring towards the impact of sodium chloride on aquatic organisms. And the third test what we have conducted, that is the experimental pesticide, is actually quinal plus. The test animals which we have selected, that is Oreochromis mozambicus. The test conducted, what are the tests we have conducted means median lethal study conducted for 96 hours, the LC50. The LC50 value we got 4.50 mg per liter. So sublethal study also conducted for 30 days at various sublethal concentration in the 1/10, 1/20 and 1/30. Here, the biochemical study and the estimation of what we call protein and lipid and the estimation of acid and alkaline phosphates in gill, liver and kidney on the fish Oreochromis mozambicus exposed to various sublethal concentration of quinalpus for 30 days observed, and the estimation of aminotransferase activity and hematological changes of RBC, WBC, hemoglobin. This also we have studied, and histopathological alteration in gill, liver, kidney of the fish Oreochromis mozambicus exposed to various sublethal concentration of quinalpus for 30 days also we have observed. Here, when we discuss about the result, the total proteins, the results showed significant decrease in the level of protein in gill, liver, and the kidney of Oreochromis mozambicus exposed to low, also medium, and high sublethal concentration of quinalpus for the period of 10, 20, and 30 days. Now, when you compare for the total protein, total lipids, here also you can see the results showed significant decreases in the levels of lipid in gills, liver, and kidney of Oreochromis exposed to low, medium, and high sublethal concentration of quinalpus for the period of 10, 20, and 30 days. Now, these are the graph. You can see here that is the total protein in the gill. See the first picture, that is here the protein level is decreased. Even the lower concentration, the medium concentration, the high concentration, among all the three concentrations, while comparing the control group, okay, all the treated groups, the protein levels are decreased. When you come for the liver, here also the same. All the three different various concentration, low and medium and high concentration, that is when comparing the control group, all the other treated groups are showing that the reduced amount of proteins. Then go for the kidney. Even kidney also you can see here, while comparing the control group, all the treated groups are showing the reduced amount of protein level. Okay. Then you can go for the lipid. That is what we have discussed. These three are protein. Now just to see the lipid. Here also the same. And you get all three concentration, lower and the middle and higher concentration, among all three concentration, while comparing with the control group, this remaining all treated groups are getting reduced to the lipid content. Then you go for the liver. That the first one is actually gill. The second one here discussing about the liver. The liver also the same. All the three concentration, while comparing with the control, all the treated groups are they are reducing they got reduced to lipid content. Okay, then you go for the kidney. Kidney also the same. Okay, comparing the control group, all the treated groups are getting reduced. So here you can see that the acid and the alkaline phosphate activities. Here also the same. Okay, for example, see the result showed acid phosphatase and alkaline phosphatase activities were increased significantly in gill, liver, and kidney of Odeochromis mozambicus. Okay, so that of control for the period of 10, 20, 10, 30, 10, 20, and 30 of different exposure. See, aspartase, then AST activity and ALT activity. Here also the same difference we can see here. The aspartate, the AST activity and ALT activities were increased significantly in gill, liver, and kidney of Odeochromis mozambicus compared to the control group. Now see here, the graph is very clearly showing that the same. Okay, while comparing the control group, all the treated groups the level of ACP is increasing. Okay, it is occurring in the gills, and even go for the liver also the same. While comparing the control group, all the treated groups, the ACP activities are increasing. Then you go for the kidney also the ACP activities are increased while comparing the control group. Then we go for the ALP activity. ALP also the same. Okay, ALP activity also, while combining the control group, all the treated groups is increasing. It is occur gill, liver, kidney, all the organisms while seeing that the ALP activities are increasing while comparing with the okay. Now the AST activity. Here also the AST activity also increasing all the gills, liver, kidney, and while combining the control group. Okay. Then ALT activity of gills, everywhere the elevations are there. It is increasing while combining the control. All the activity, all the treated groups, this activity is increasing. Now, when you come for the histological work, when you discuss about the histological study, here while combining the control, the treated groups, so we can see a lot of changes. For example, we can observe some mucus secretions, vacuolation, and necrosis, and fusion of secondary lamellae. Okay, so these are the variations we can observe here in the treated groups of gills. Okay, now while discussing about the biochemical result or biochemical estimation, the total protein, while discussing about the total protein, a reduction in the protein content in the present investigation in Odeochromis suggests that the tissue protein undergoes proteolysis, which result in an increase in the production of the amino acids. Are utilized for energy production during stressful situations in the intoxicated tissues. Total lipid. When we discuss about the total lipid, a reduction in the lipid content in the present investigation in Odeochromis may be due to liver dysfunction or mobilization of glycerol or inhibition of oxidative phosphorylation. Lipids serve as energy source for fish metabolism and hence reveal their importance during stress condition. Decrease in tissue lipid indicates that lipid hydrolysis might be active to derive energy to overcome toxic stress. Then, phosphatase activity. We discuss about the phosphatase activity. Here, ACP and the ELP, we have to see that the increased level of ACP enzyme activity at all the concentration of phenolphos might be due to the increase in protease activity, which caused damage to the lysosomal membrane, thus permitting the leakage of lysosomal enzymes into cytoplasm. Changes in the enzyme activity are due to adverse effect of xenobiotics on the cell and its organelles. Increased activity of ALP in blood plasma may be related to hepatic tissue damage and dysfunction due to pesticide toxicity. The elevation in ALP activity may be due to an increase in transphosphorylation activity. Now come to the histological study. When you discuss about the gill, the fish Oreochromis, when exposed to sublethal concentration of phenolphos for 10, 20, and 30 days, showed marked histopathological changes in the gills, such as excessive secretion of mucus in the intercellular space and the fusion of secondary gill lamellae, reduction in length, swelling of epithelial cells of secondary lamellae, swelling of primary lamellae, damaged RBC cells, curling of secondary gill lamellae, rupturing of secondary gill lamellae, cytoplasmic vacuolization, and necrosis. So, like that, a lot of observation we got from this. And when you come for the conclusion, actually what we want to say that the present investigation concluded that phenolphos induced a biochemical, enzymological, hematological, and histological alterations and caused a significant metabolic effect on the physiological consequences. These parameters offer a rapid and sensitive means of monitoring towards the impact of phenolphos on aquatic organisms. Now, the next test what we have done, that is the impact of insecticide endosulfan. This is a very old test we have. When the endosulfan was burned, before that we have conducted this test. Here that we have taken the experimental pesticide endosulfan, that is a test animal we have taken as a Chenna striatus. Now the test conducted, that is a median lethal study conducted for 96 hours and sublethal study conducted for 20 to 21 days at various sublethal concentration, and a lot of work we have done. But I have constructed some work here, and I'm going to show some work only here, and that is FTAR and the STS page and some work. So one or three or four, 2-3 works only I am going to show. Then see, that is the present study was carried out to analyze the toxic effects of endosulfan in the gill and the liver tissues of freshwater fish and striatus by using FTAR spectroscopy. See the picture here. Here, the table, the FTR gill, regarding that, the very clear picture is given. The control 7 days, 14 days, and 21 days. FTR study graph is represented here. This graph, what the graph says, regarding the gills, see that the present 21-day study, the figure on the table shows that compositional structure and the functional changes of amide A engage stretching of proteins. CS2, asymmetrical stretching of lipids. CS2, stretching of lipids. And amide I, and stretching of proteins. Aromatic, stretching in nucleic acid and proteins. These are the significant wave number difference and bands area values between all groups and gills. Then we go for the liver. Liver also we can see that changes here. In the present study, the figure on the table shows that compositional structure and functional changes of amide A, NH, stretching of the proteins. Okay, 32972, 3302 CF. CS2 asymmetrical stretching of lipids, is to stretching of lipids, amide I, and the CO stretching of proteins. Aromatic stretching is stretching in nucleic acid and proteins. There are significant significant number differences and band area values between all groups and liver. Okay, so this two, with this two, repair of denatured proteins. The present investigation reveals some alteration in protein fractions were found due to toxifications. The gel electroprogram obtained from gill and liver showed marked alteration in the protein fractions and profile. Toxic substances causes acute, chronic, and latent from the disease depending upon the subsolubility of the fish, mode of administration, and the duration of exposure. Physiological status of the fish may contribute to the biological profile of infection. Here, the stage page that work also we have done. Here, the protein profile analysis we have made. The electrophoresis of protein is an effective technique for generating systematic data from macromolecules. Electrophoresis cannot demonstrate the identity of two proteins and only show differences. STS page is a technique widely used in biochemistry, forensics, genetics, and molecular biology to separate proteins according to their electrophoretic property. Now, this is the binding pattern of gills. You can see here also we have that the organisms are treated with various days. For example, 7, 14, and 21 days. And the gill tissues collected from the organisms. So we have sent the tissue for the binding pattern. Here we have got the output that is the electroprogram of the gill of control fish of Channa striatus figure 1 and the protein fraction in the lane 1 are listed in table 1. Here very clearly we have mentioned that it refers to 8 protein fractions are namely 118, 106, 85, 67, 63, 56, 44, 44 and 7 kDa. And lane 2 refers to 7 days treated Channa striatus with the sublethal concentration of endosulfan. It shows 8 protein fractions, namely 103, 83, so and so. And the electroprogram of the lane 3 represent the gill of Channa striata treated with the sublethal concentration of endosulfan for 14 days of duration, and it exceeds 8 protein fractions, namely 103, 87, 72, 61, 45, and 26, 6, and 4 kDa. The lane 4 exhibits the 21 days treated gill Channa striatus. It shows 8 protein fractions. So namely 90, 69, 62, 51, 42, 32, 18, and 8 kDa. So when you go for the banding pattern of liver, here also we can see there is the lane 1, the lane 14 is given the protein fraction. The protein fraction in the lane 5, okay, are listed. It shows 4 protein fractions, namely 72, 73, 59, 47, and 26. Lane 6 refers the 7 days treated Channa striatus with the sublethal concentration of endosulfan. Okay, it shows 8 protein fractions, namely 1 and 2, 8, 5, 76, 5, 57, 51, 39, 31, and 19 kDa. And lane 7 represents the 14 days treated Channa striatus with the sublethal concentration of endosulfan. It demonstrates 5 protein fractions. And lane 8 is showing the 8 protein fractions. So these are the fractions we have observed. From this, what discussion we have done means the gel electroprogram obtained from gill and liver showed marked alteration in the protein fractions and profile. Toxic substances cause acute chronic disease depending upon the subsolubility of the fish, mode of administration, and the duration of exposure. Physiological status of the fish may contribute to the biological profile of effectivity of disease causing pathogens, chemical substances in tissue, cellular, and molecular level. Now we have made work on this some study, scanning electron microscope examination also we have made it in the gill. So here, while seeing that, while comparing the control, the treated group, we have observed a lot of changes. The same degeneration of secondary gill lamellae and bacterial formation, so fusion of secondary gamellae. So, number of variation we have observed here. Okay, that that will be here. That's when the fish is exposed to the right sub-lethal concentration of endossulfan, it shows the thickness of the gill filaments, increase in the mucus secretion, and the surface of the gill filament secretion on the surface of the gill filament, loss of micro ridges, okay, and the numerous micropores appeared on the gill surfaces. So these are the observation we observed. Okay, so from this, we have to we are coming to the conclusion that some care should be taken before buying some chemicals and pesticide or whatever it may be, insecticide, and some regulations should be followed. For example, if you see the bottles of the what you call these toxic substances, here some label will be used. The label we have to check everything is there or not. For example, the pesticide and the chemical label consists of that a binding legal agreement, whether it is there or not, we have to check. Then trade name and amount of active ingredients, and directions for use, toxicity ratings, registration number and establishment number, name and address of the manufacturer, and net contents, the signal word, for example, danger, warning, caution. Okay, so these are the information should be present on the label. Whether it is there or not, we have to check. And how to reduce this risk? There's a better use a pesticide and the chemicals only when necessary, and use less toxic pesticides and chemicals. Use safe, sensible application method. Okay, conclusion. From this, we are concluding usage of pesticide, fertilizers, and chemicals cause damage to the aquatic environment when it is used in above the recommended level. So we request all of you to use, while using the chemical, while using the fertilizers, whatever it may be, maybe it may give the benefit to us, in the agriculture side or some other activity for killing the insect, for killing the pest, whatever it may be. But while using, that should be used to the recommended level. You should not use it beyond the recommended level. When you go for the when you use beyond the recommended level, the number of causes you may get. The causes not only occurred for aquatic living organism, from the aquatic living organism, it is passing to the human being also. We also getting number of causes and a number of problems. Okay, number of diseases also we are getting. Even our lifespan also reduced because of these toxic chemicals. So from this, I'm concluding my talk. I'm very thankful to the organizers and the organizing committee and the convener of this activity. Thank you very much. And once again, if any questions, please, if any questions, please mail me. The mail address I have given here is leon435@gmail.com. Okay, thank you very much.
Thank you, sir.
So,
Thank you, sir.
thank you, Ma. Thank you.
Thank you, sir, for your exemplary speech. So now it is the time for the queries. Participants, you can post your questions in the chat box. Sir, shall I read the question, sir? Sir?
Yes, Ma. Yes.
What are the impacts of micro and nanoplastics on aquatic biota and ecosystem?
Hello, I couldn't hear, Ma.
Sir, what are the impacts of micro and nanoplastics on the aquatic biota and ecosystem?
Yeah, of course. Actually, the normal, if you see the normal toxic which we are using, for example, insecticide, pesticides, these also giving lot of changes in the aquatic biota, like plastics also. Okay, for example, if you, before going to the aquatic biota, just you take the organism which is living here in the terrestrial mode of life. For example, cow. Okay, when the cow is taking this aquatic, the plastic, sometimes they are having along with the food materials. Okay, they are getting some stomach disorder. Like that, though, when you this plastic substances when it is reached to the aquatic media, okay, that is not only sometimes if it is very small, it is consumed by some organisms and they are getting affected. Not only that, even this bioplastics and bioplastics are polluting the environment. Polluting the environment. They block the digestive tracts of the living organisms. Okay, and also it is the stuff, the plastics. Some plastics actually what making the organism to die. Also even. Okay, not only that, if the plastics are dumped into the aquatic media, the living organism which is living in the aquatic media, they cannot live happily. Okay, because they are getting infected. They are getting infected because the living organisms, the aquatic media, for example, microplastic particles can physically damage the organs and leach hazardous chemicals from the hormones distributing distributing inside the aquatic media. Okay, and even they are affecting the immune system of the living organisms also. Okay, microplastics in the water we drink and the air we breathe can also hit a human directly. Okay, so it's very dangerous. It is very dangerous. Not only aquatic organisms, if indirectly it is affecting the living organism living in the terrestrial mode of life also.
Okay, sir. Thank you, sir. What is the impact of sulfate on algal community?
Impact of
Sulfate. Sulfate on algal community.
impact of sulfate on algal community. Yeah.
Yes,
Okay,
sir. Yes, sir.
okay. Sulfate actually, the availability of sulfate also has been repercussions on the overall metabolism of algal cells because of its many pivotal roles in cell physiology. Okay, the maintenance of homeostasis and the responses to sulfate deprivations have a strong impact on photosynthesis. Mainly, it is affecting the photosynthesis. Okay, and carbon, nitrogen. Okay, and all the metabolic activities also getting affected when the sulfate is taken by these algae. Okay, not only if the other organisms are when the organisms are consuming the algae. Okay, the organisms also ultimately getting the problem because of the sulfate. Okay.
Thank you, sir. Next question is, what are the heavy metals found in fish?
Pardon? What are the
What
heavy
are the
metals?
heavy metals found in fish?
What are the heavy metals are found in fish?
Fish.
Okay. Just before now, while taking the class itself, I said, okay, number of heavy metals. For example, zinc, okay, cadmium, chlorine, sorry, and the copper, all everything. What are the chemicals which is mixed with the water bodies? Okay, this is reaching into the aquatic media through aquatic media. It is entering into the fish body. Okay, for example, everywhere, not only this copper, zinc, and a number of metals are there, which is mixing with the aquatic media. This directly is entering into the aquatic organisms and it disturbing the life of the aquatic organisms. Okay, anything more?
Okay, sir. Thank you, sir. Thank you, sir, for your patient clarification of doubts. Shall we validate the session, sir?
Okay, ma'am. Thank you. Once again, I say thanks to all the team, all the team members, and the convener of this program, and my special thanks to the organizer, or the organizer, Dr. Krishnaveri Madam. Okay, thank you one and all. Thank you very much. Thank you.
Thank you, sir. Now we are in the end of the session. I invite Mrs. P. Philomena Mary, Assistant Professor in Microbiology Department, to propose the vote of thanks.
Good evening to all. Gratitude opens the door to the power, the wisdom, the creativity of the universe. I thank our God Almighty who showered his blessing in this occasion. An honorable man restores the dignity of others. I thank our principal, Reverend Sister Dr. Amala, who has been supporting continuously for us. I thank our college secretary, Reverend Sister Gladyn Arokya Mary, our vice principal, Reverend Sister Dr. Vimali Mary, and our administrator, Reverend Sister Anthony Amal, who have been supporting in various ways. Gracious words are a honeycomb, sweet to the and healing to the bones. I thank our resource person of today's section, Dr. Prakash Shagaya Leon, for his inspiring speech on the topic Toxicological Impact on Aquatic Biology. Thank you, sir. Your speech takes your hand, opens your minds, touches your heart, shape the future. Thank you very much, sir. I consider it a great privilege to propose a vote of thanks to all the participants for your cooperation in making this FDB a memorable success. I extend my sincere thanks to our HOD, who is the director of this FDP, for efforts, and all the faculties of our department for the hard work and support. I would like to extend a special thanks to Mr. Magesh Shah for his technical support. Once again, I thank one and all to this wonderful occasion. Thank you.